Hi, I'm Devin Lindsay.
I'm Logan Groves.
I'm Andrew Reiner.
I'm Alex Klom, and we're all in period 4.
In this experiment, we used the RNAi pathway in C. elegans in order to silence a gene. By silencing this gene, we are able to tell what the function of the protein coded for by the gene is. We perform this by chunking N2 wild-type C. elegans onto a plate containing RNAi bacteria. These RNAi bacteria were modified so they produce double-stranded RNA for the Rol6 gene. We then let our C. elegans eat these bacteria. When they consumed the bacteria, they got the double-stranded RNA in their cells, which would hopefully trigger the RNAi system in our C. elegans cells.
The wild-type worms demonstrate movement characterized by swimming like a slithery, snaky snake in a sinusoidal wave-like pattern. The eggs are clearly visible and ovular-shaped. The L1 stage has tiny little guys with skinny and short bodies and looks like they could curl up to fit inside the same size as the egg. The L2 stage is represented by worms that are a tad longer and slightly thicker than the L1, but are still very cute little noodles. This trend of coming to age continues through the L3, where they continue to eat and grow fatter and longer. The C. elegans become L4 stage as they begin to show characteristics of adulthood, but are still definitely smaller than adults. Once adults, the worms are very thick and large, but visible eggs are seen inside their bodies.
Here, you can clearly see that our attempt to induce the Rol6 gene in C. elegans has failed due to the fact that the C. elegans are still swimming in their typical wild-type sinusoidal motion. In this video, you can see what the C. elegans should look like when the Rol6 gene has been turned off by the RNAi pathway. The worms should be swimming in a C shape rather than their wild-type sinusoidal shape.
We tried to silence the Rol6. This gene is one that encodes for a collagen protein that is contractile. The protein encoded for by this gene interacts with the protein encoded for by a gene called SQT1. These two proteins act similarly to antagonist muscles by contracting against each other to create movement. So in the RNAi pathway, to silence the Rol6 gene stops that protein from being translated. Only the SQT1 gene is being expressed, and instead of the wild-type sinusoidal movement, they move in a right-handed circle. The RNAi pathway starts immediately when the dsRNA for the Rol6 gene enters the cells of the worms. In this experiment, we performed this by feeding the C. elegans RNAi bacteria that had been previously modified to produce the dsRNA we needed. Once the dsRNA enters the cell, the dicer, which is an RNA protein complex, cleaves it into shorter segments called siRNA. These siRNA bind to multiple proteins, forming a complex called RISC. Within this complex, the siRNA is unzipped. The RISC then probes the cell for the matching mRNA, which once it finds, it cleaves so it can't be translated and is effectively silenced. This is what we tried to do with the Rol6 gene in our C. elegans.
Failed. We were unable to induce the RNAi pathway to shut down the Rol6 gene, but it was still overall a great experiment, and we had a lot of fun working with the model organism C. elegans.
It's really great to have this experience with an organism that we might use further in college and future studies.